It has become customary to conceptualize the living cell as an intricate piece of machinery, different to a man made machine only in terms of its superior complexity. This familiar understanding grounds the conviction that a cell's organization can be explained reductionistically, as well as the idea that its molecular pathways can be construed as deterministic circuits. That's another uh,
Keyword.
keyword, deterministic. The machine conception of the cell owes a great deal of its success to the methods traditionally used in molecular biology. However, the recent introduction of novel experimental techniques capable of tracking individual molecules within cells in real time is leading to the rapid accumulation of data that are inconsistent with an engineering view of the cell. This paper examines four major domains of current research in which the challenges to the machine conception of the cell are particularly pronounced: cellular architecture, protein complexes, intracellular transport, and cellular behavior. It argues that a new theoretical understanding of the cell is emerging from the study of these phenomena, which emphasizes the dynamic, self organizing nature of its constitution, the fluidity and plasticity of its components, and the stochasticity. I think I said that wrong.
Stochasticity.
Okay, we're gonna one of these days learn how to pronounce that, but you know what I mean. And nonlinearity of its underlying processes.
Okay.
Okay.
Okay, so the paper roughly has four sections.
Mhm.
The first section here, however, our progress in translating this ever-growing repository of information into a deeper theoretical understanding of what living systems are and how they function as coordinated wholes has been far less impressive. So, past half a century, molecular biology has generated vast amounts of knowledge, but our ability to translate this is less impressive, is what he's saying. He's basically complaining that we haven't made as much sense of the field as we could if we had a different paradigm. So this is about a paradigm shift in biology that has been growing, I think, in since 1990. It depends on which biologist you talk to, and of course, scientists like to fight with each other, and the old ideas are ushered out very slowly. I think Dr. Dan or somebody said something about, or no, what was the doctor? No, it was R.J. Downard said that it's like science progresses one obituary at a time.
Oh yeah. It's because these paradigms, at least the way it was outlined by Thomas Kuhn in that 1962 book, The Structure of a Scientific Revolution, is that these paradigms become these images, almost symbolic, of how a particular concept works, and people become, or scientists become, almost attached to them. I don't want to say religiously because I don't want to alienate the entire scientific community, but they become rather committed to them, especially if they're the ones that were in on the early days of that particular paradigm, the research coming off of that paradigm, and then to make people go from an old way of looking at things to a new way of looking at things because of natural human processes. We just don't like to change sometimes.
Right.
And of course, we've seen this in our, shall we say, at our opposites on the creationist side seem to be very much in love with this machine concept of the cell, because they cling to a teleological view of the universe, that things have been created with us in mind and with for a particular purpose.
Right.
And that's what all this love of the mechanistic view is an excuse to say God did it. But if the machine view of a cell or of biology doesn't hold up much anymore, then what does that say about all the stock you've been placing in that to prove you're God?
Yeah, and the interesting thing is that they are constantly, they constantly give us the rhetoric that they are the ones that are on the leading edge of science.
Mhm.
And the leading edge of science for them is finding more and more complexity in the cell.
Mhm.
And they like their minds are always blown. My mind is blown. They use that, well, particularly Matt Ass uses that over and over again. Oh, my mind was blown when I found this new complex thing. Well, what they're finding and what'll be shown in this paper or talked about in this paper is that there are so many different mechanisms all happening at the same time. We keep unwinding more and more and more of them, but it has turned into more of a zoo than a machine. So,
right. And, and I'm not saying, I don't want to create too much of a dichotomy here. The machine view or the deterministic view of the cell or of biology in general, it's not that it wasn't true. It was true in a sense at the time, but now it's not as useful. We're losing information or the opportunity to gain new information by clinging to that mechanistic view of things. And so switching to a more whatever that word is,
stochastic.
I can't believe I can't pronounce that anymore.
Just stochastic is good enough.
Yeah, to a more stochastic view of and dynamic view of the cell, you will gain information plus show how, more completely and more adequately how the concepts under the mechanistic view of the cell, how those items really work.
Yeah.
And so even the stochastic view is true in a sense. And that's the one thing I think that sometimes people get uncomfortable with. The truth that you're seeking, these models only take you so far. And if you don't know when to let go of them and adopt a different model of the same thing, then you'll cease gaining knowledge.
Yep. The main interpretive framework in molecular biology is mechanicism.
Mechanicism, I would call that.
Mechanicism, a highly influential research program of many forms and incarnations that can be traced all the way back to the natural philosophy that gave rise to the scientific revolution. And here is the late immutable destiny.
Immutable.
Hey, what's going on? I told you I was going to be late, Michael. I texted you about this.
The late immutable destiny.
Oh, Michael.
I was busy, so I wasn't reading texts.
Well, okay, it's not my fault you didn't read my message that I preempted you with.
I don't know what you're talking about. Modern proponents of mechanicism conceive of the cell as an intricate piece of machinery whose organization reflects a pre-existing design. And then they call that the machine conception of the cell. The MCC long predates the rise of molecular biology. Its history runs parallel to that of mechanicism, which is why one can find rudimentary expressions of the MCC dating back to the 17th century, when analogies between machines and organisms first began to acquire currency. Mel Pidgey, one of the founders of microscopical anatomy, attributed the bodily functions of animals and plants to a very large number of machines, which are of necessity made up of extremely minute parts, invisible to the naked eye. So that is, I'm not sure when that man existed, but it sounds like a long time ago. So, and what's that?
What page are we on?
We're on page 2 or 109. Are you, do you have the paper and you're reading along?
Yeah, I do. Let's see.
Yeah. And I could probably make this. Could I make this paper bigger by doing
zooming?
That? Yeah, by zooming a little bit. And
yeah, we just don't want to lose resolution, but it looks good to me. But I've got it on full screen.
Okay. I think that might be a little bit better. Hey, well, let's see. This. Okay, now I'm trying to. Unfortunately, okay, they call it a small chemical laboratory. That's one of the ways of depicting it. And
or a miniature factory.
Yeah, miniature factory.
that's not exactly like a bad description of kind of what cells do on a conceptual level, because nature has figured out how to transform certain compounds that have certain functional groups, so groups that are have specific kind of reactivity, and transform it into another kind of chemical that has different reactivity.
Yeah, and he explains why some of that
Yeah,
breaks down a little bit later.
I think it's different in the sense that when you study how the machines actually well, if you want to call enzymes machines, like how these actually work, it's very it's very non-traditional to the way you would think about something being constructed. Like if you were wanting to construct like I don't know, like a wheel, or like some kind of like metal object or something like that.
Yep.
Because chemical transformations operate kind of on the basis of this kind of parent molecule entering into this enzyme, adopting some conformation while it's bound to the enzyme, and during that bound state, uh, something within the enzyme, something within the active site, permits for part of that enzyme to react with the substrate and to change it in some specific type of way.
Yep.
Where machines don't seem to do that type of thing in the way that usually you define something where a machine will you define, for instance, like if I want to cut off a specific length of wood, right? I make a demarcation, or I make a laser with that's thin enough to be able to do that, right? It's there's like an aspect of determinism that kind of gets accommodated with the process of
Right. Determinism and being more precise are kind of the hallmarks of machine design. We
yeah.
design machines to take input and produce a product, basically.
Right. I mean, what a machine is, and correct me if I'm wrong, because I'm not, a machine designer or anything like that. But to me, what a machine seems to do is you give a machine some kind of input, right? And by virtue of some rules that you've defined for how it's to process that information, and for how it's to perform certain operations on that input, it gives you back some result. So essentially you have some input, some, if you will, some kind of filtering device that operates by a set of rules, and then you have the output, whatever that machine happens to output as the end result of your giving it an input.
Yep.
And it's very deterministic in that way, right? You give if you provide the same input to a program, or to something that that's operating under a prefix set of rules, it will always return the same result, right? Whereas with something like a cell, for instance, and especially in the case of like proteins don't always they aren't perfect in terms of their ability to catalyze. They're very good at it, right? But they never are able to catalyze all the all of the substrates that are present. And the thing is too, if you consider uh, something else, so proteins, especially like drug targets, for instance, um, you you design, essentially what you're doing is you're designing a molecule that's in the likeness of a molecule that the protein knows how to deal with. That the protein is used to seeing some molecule on a day-to-day basis, and it already is, you know, perfectly fine with transforming A to B. But when you introduce something, very, 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 very similar, right? You change something about, maybe there's a methyl group, one carbon to the right, or whatever it happens to be. It can be a very small change in terms of in terms of the actual chemical structure, and you get radically different results from it. You get sometimes no reaction, sometimes you get transformation into a product that's very toxic, or sometimes there's something about that reaction trying to proceed, and essentially it just causes this molecular machine to kind of break down. It doesn't function anymore, despite there being a high level of similarity in the types of structures that it should be able to recognize.
Okay, moving on. Let's see. Following the Second World War, which is kind of interesting. So this started very early. The pioneering ideas of cybernetics, information theory, and computer science captured the imagination of biologists, providing a new vision of the MCC that was translated into a highly successful experimental research program, which came to be known as molecular biology. So that's kind of where it started after the Second World War. And we had a lot of the ideas of, I don't think we'll have time to read the entire paper, so I think I should just go with the highlights. So,
well, I mean, we can go through the entire thing. I mean, I just don't know how long.
Yeah, it would be far too long for the average reader. They could always read along or pause and read the pieces that they want to catch. But at its core was the idea of the computer, software and hardware, and that kind of stuck. And when you think about cells and the mind, for that instance, or neuroscience, the demarcation between the hardware and the software disappears completely. Hardware is the software, software is the hardware. There's no way to draw that line very well in biology, but he kind of gets into that.
Yeah, because what machine or assembly line contains within itself a blueprint?
Right, a completely automatic
external. But is that a true statement? Am I being accurate that the cell contains its own blueprint and follows its own?
Well, to a point.
Blueprint.
I think he kind of pushes back on that a little bit later in the paper too.
Yeah.
A completely automatic factory for fabricating automatic factories like itself.
Yeah.
That's one thing that's different between factories and living things, is that this thing creates copies of itself that are reasonably pure copies, but not totally, and that's where we get an evolution. So
right.
I think
to push back on that idea, right? I mean, there is no reason, like, inherently why a machine couldn't replicate itself, right? I mean, there's nothing forbidding you programming a type of machine that within the code of that machine, within its heart, right, it has the information needed to make a copy of itself, right?
Yeah, we could make machines like that. We just haven't pulled it off yet. Oh, let's see. Our basic theoretic picture of the cell since 1962 has essentially remained unchanged. The standard view nowadays is that cell coordinates its functions by virtue of a genetic program encoded in DNA that directs, and that's kind of the standard model, that directs and controls the expression of a specific set of RNAs and proteins, which assemble deterministically into stable molecular machines. And there we go again, that reliably and efficiently execute predetermined operations according to mechanisms of cell division, endocytosis, signal transduction, etc. Machine analogies and metaphorical references to locks, keys, gates, pumps, motors, and engines continue to pervade the technical literature. And this is, he's assessing where we kind of are, except for a relatively fringe group of biologists that are kind of charging off on their own, and some problems that have developed, which he will detail a little bit later. let's see, what else do we have here? Does anybody want to read the paper? But why?
I was noticing this, the next paragraph. What's so special about machines that make them such opposite analogs for thinking about cells? Although there are many different kinds of machines, a machine can be characterized in very general terms as a device with fixed interacting parts that operate in a coordinated fashion to produce a predetermined outcome. More specifically, one can identify four distinctive properties of machines that are particularly relevant in the contemporary formulations of the MCC. First, machines can be described in terms of a list of parts and a blueprint indicating how those parts fit together. Oh, I just made a weird mistake. Meaning that someone who has never seen a particular kind of machine should, in principle, be able to assemble any number of copies, each virtually identical in appearance and performance, provided they can consult the machine's design specifications. Second, as machines are designed to perform highly specific functions, their operation is tightly constrained, which is why it is possible to predict and control their behavior. Third, machines are highly efficient in what they do because they always follow the exact same sequence of steps in every cycle of their operation. And fourth, the operation of machines is not continuous. Their functioning can be interrupted and their parts examined without thereby jeopardizing their structural integrity. The first and fourth of these characteristics account for why the MCC justifies the belief in the sufficiency of reductionistic explanations of cellular phenomena, whereas the second and third show that the MCC provides support for a deterministic view of cellular processes.
Yep.
So that's his the definition of machine, which is really important because when you're
Yeah.
when
Do we want to go through?
I'm sorry, go ahead.
Do we want to go through each of those criteria? Because hearing that and kind of and listening to that, I do have I do have my own perspective on it, but I need to find that again because I just lost track of it. Where and how?
Kind of what he does in the paper here a little further on.
Where is it? The list? Let's see.
This is page 109, 110, something like that.
Okay, one sec.
In recent years, the MCC has come under attack from various fronts. Let's see, what do I have as a result? As a result, critical reviews have begun to appear that explicitly challenge the reductionistic and deterministic presuppositions of mechanism and question the coherence of the familiar clockwork image of the cell. Did I say clockwork?
Clockwise, yeah.
Like clockwork.
Clockwork. Being exceptionally British today, Speeder. You know that?
Clockwork. Anyway, and then they list a whole bunch of papers and let's see, drawing and building on this burgeoning body of literature. The aim of this paper is to establish the inadequacy of the MCC. From a theoretical perspective, the MCC offers a poor and rather misleading representation of biological reality, or so I will argue. And there we go. What else we got? The first.
Well, in the next one, he brings up the idea of self-organizing rather than self-assembly, and then the thermodynamic equilibrium.
Yep.
And then he brings up the fact that, hey, guess what? Molecules are small, and so the forces that act upon them are different from, the way forces act upon a machine at the macro level. And that has to do with the Brownian motion that he's going to make a lot of devote a lot of words to, and then even quantum.
Well, the thing is, right? It's not that the forces necessarily are different. I mean, the forces that act on a protein, for instance, are the same that act like if you were to compress to compress a fluid or to compress a metal sheet or something like that, right? What is different though is that compared to something macroscopic like a like a ball or a rod or something like that, microscopic objects, especially when you get to like very small scales, on the scales of like atoms and molecules, it's the case that quantum mechanics is the language there, and the one of the important aspects of that is determinism just completely fails. You have to describe things in terms of probability, um, and that kind of and that type of framework, it really changes the types of ways you think about how chemical reactions or certain interactions of systems that are macroscopic work, even if they're made of even if they're made of small subunits. Um, so yeah, I mean, it's not the fact that the laws are different; it's the fact that the way that these macroscopic objects are made, or the parts they're made of, being microscopic, right? Parts of that structure, parts of the microscopic parts of that structure, are bound by a different set of rules than that govern ordinary objects we
Right.
normally encounter, like a ball or
Yeah,
uh,
for one thing, they're flipping conformations at about 60,000 miles an hour, and they're
yeah,
they're
it's insane.
constantly
I mean,
in flux.
Well, what's the level of speed again?
I don't know if I'm just throwing that out as a
Oh,
the,
okay.
yeah.
the thing is, right? It doesn't. You could put a number to it, right? I'm sure you could compute on average how many times there's fluctuations within the vibrations of some atoms or whatever. But the important thing is that it's such a large number. It doesn't make sense to really try to really try to put it into words. It's insanely large.
I think, let's see, the important part of this paragraph, that cells are self-organizing fluid systems that maintain themselves in steady state far from the thermodynamic equilibrium by continuously exchanging energy and matter with the surroundings. So you got to think of them as very dynamic states. That's sort
Speaking
of
of fluid systems, here's a kind of a question. Would you machine parts are not surrounded by a liquid, and factories aren't filled with liquid. Like maybe some are, I don't know. And with things operating, with the parts operating inside that liquid, how thick? What would be a good analogy to describe how thick the fluids inside a cell are? Like if we were walking around inside a cell, would it be like walking through a swimming pool? Would it be like walking through maple syrup? What kind of
I think
environment?
Vaseline. I think like walking through a swimming pool filled with Vaseline,
Uh huh.
petroleum jelly,
Uh
that
huh.
sort of thing. It's pretty thick, and it's packed with
Okay.
water and proteins to the point where, if you look at the radius of the average protein, there's another protein within less than that radius next to it. So that's how packed it is with proteins.
Wow.
So,
Okay.
so it'd be like walking. So the Vaseline at that level behaves, or the water behaves kind of like petroleum jelly. So it's real thick, or like thick grease,
Yeah.
and studded in there are these a ton of proteins and RNAs that are very, very close to each other, and they're all moving rather quickly through that mush, that sort of thing.
Wow. Okay.
And yeah, so it's more like a cheese dish, maybe some Alfredo. It's there's
I love Alfredo.
I love Alfredo. I gotta go make some of that tonight.
I'm gonna quit my job and become a biologist just because you said Alfredo.
Okay.
Sorry, I was dreaming.
Yeah. Anyway, yeah, the water is fairly thick at that size. I have heard, and the water actually has an effect on folding proteins and a number of other things. So I mean, it's very difficult to look at this from the machine point of view and understand what is really going on inside the cell. And I think that's what this whole paper is about. So anyway, Let's
Well, that's
see.
very interesting. I didn't realize it was that thick. So
Well, it's pretty well packed, and I don't know, mutable. You had a comment about how fast, but the thing is, things are flittering around very rapidly. Like the
mhm.
water molecules are flipping back and forth, but the things are bumping into each other constantly at that level. The water is fairly tiny compared to the protein, so
Sorry, what was the what were you guys? I'm sorry. Somebody was asking questions, and I had to.
I was just kind of wondering about the relative thickness of the liquid inside a cell. Like, is it
Yeah,
more like water or maple syrup or grease, like petroleum jelly? Like,
I think it'd be akin to if you like when you have kids and you like you mix fluids of different densities together. I think that'd kind of be what it'd be akin to, because for instance there's certain there's certain parts of a cell, like the lipid bilayer, for instance, right? Um, Lipids are very oily. They're not very easy to. It's not exceptionally easy to get something to go in between them, right? If you move one of them, you move all of them, really, because they once you put more than one lipid molecule in have more than one lipid molecule inside your vesicle of whatever kind, it starts to behave as one system. The chemical interactions there kind of cause it to be very what's called homophilic.
it'd be almost like wax-like.
Yeah.
Yet it floats around. Yet it's also like the liquid on a bubble, because it does float around, and the transmembrane proteins float around on it like rafts, and they float around fairly fast because some mechanisms require that two of those transmembrane uh, proteins bump into each other and connect.
Well,
So
and,
I mean,
here's
even
the
that's floating around very quickly.
and here's the thing too, right? And this is a very quick point. The reason the reason certain proteins are embedded in lipid membranes is one of the one of the codes, so to speak, for son of a bitch. One of the codes that tells proteins where they're going to be sent and where they're going to be placed for for the cell's functions has to do with a sequence of it's like 6 or 7, I believe, hydrophobic residues. What are hydrophobic residues? Well, hydro is water, phobic meaning scared of, but chemists use it to mean hating. Um, so you have in some protein, right? You have a domain or a sequence of amino acids that are really, really, really not attracted to water, and you have them strung together in a row. So these things are really trying to avoid, being touched by water if they can. And it turns out that the types of amino acids, the types of hydrophobic amino acids that are present and used for that kind of signaling process of, oh, you have these residues, you're going to go into the lipid membrane space. It turns out that it's hydrophobic enough to be integrated into the structure of the lipid membrane, because the types of properties that small little sequence has is actually very attractive and very synergistic in a way to the types of properties that lipid membranes inherently have. If that made sense.
Okay.
It sure does.
Okay, this is a pretty important paragraph here. If the facts that underline the inadequacy of the MCC really are indisputable, why is it taking so long for us to start taking serious notice of them? I suspect that part of the answer has to do with the resistance that many biologists intuitively feel towards denunciation of mechanism and perceive. And he goes on, let's see. But an even more important factor, I believe, is that we have been blinded by the traditional biochemical and biophysical methods of studying the cells. Until relatively recently, it was only possible to examine the cell's interior with crude in vitro techniques, looking at that means outside of life in a laboratory dish, looking at average behaviors of large populations of macromolecules under conditions usually remote from those existing in the cell. Vitro, I believe, means glass. So that literally means in glass, whereas in vivo is in life. So we're looking at them in glass, not in life. And then the introduction of novel methods capable of tracking and manipulating individual molecules within cells has allowed us to observe for the first time the real-time dynamics of biological macromolecules and the surprisingly wide range of behavioral repertoires they exhibit in vivo conditions, so in life. And as I will discuss in more detail later, single molecule studies are yielding results not anticipated by the use of population average methods. So when they do this, when they do this in vitro, they use population averaging methods and they come up with how they think the mechanism works. But when they look at individual cells, they're finding a great range of variation in these individual molecules. And he gets into a little bit of a discussion about the reality of protein folding a little later, which is kind of cool.
I mean, is that unexpected though? I mean, for instance, right, in a laboratory setting, right, you have a set of very controlled parameters that you can manipulate to a very high level of your desire. You can perform the experiment, as many times as you desire to get accurate data and perform analysis, and okay, fine, that's great. But within a cell, though, right, no cell experiences the same amount of inputs as any other cell around it, right? There's always some level of variation in terms of what. I mean, my criticism here is that should that not be to some extent expected? I mean, even in physical settings, if you change things slightly, right, especially conditions that are sensitive to small quantities, then yeah, you're going to expect to some extent that there's some change, or sometimes even drastic change in the types of behavior your system exhibits.
He mentions this later in terms of context. That context is very, very important in the operation of every cell, and every cell has a unique context. There are no two cells that are doing the same thing in any tissue anywhere. So it kind of gets into that. As more molecular biologists are becoming aware of the numerous problems plaguing the MCC, this paper will examine in detail 4 specific domains of research where the incompatibilities with the MCC are becoming particularly pronounced. So, and I think we can jump ahead to my next highlight. Let's see, as I will show, how far do I go? Does somebody else want to read this paragraph?
Sure. As I will show, according to this alternative view, the cellular architecture is regarded as a fluid, self-organizing process. Protein complexes are considered to be transient, pleomorphic ensembles. Intracellular transport is deemed to result from the harnessing of Brownian motion, and cellular behavior is viewed as a probabilistic affair, subject to constant stochastic fluctuations.
And that pretty much sums up the whole paper.
Yeah.
Let's see. So pleomorphic ensembles for proteins means that they have more than one shape and they often have more than one function. So the idea that a protein is created, and according to, should I mention them? Nephilim Free, if one amino acid changes, the whole machine breaks down, was totally, completely false. They not only...
We knew that was false for a while.
Not only can there be a lot of modification and still end up with the same function, but one protein can fold in more than one way and create multiple functions. hence the word pleomorphic, which means many shapes, that sort of thing.
I have a couple of questions.
This is yeah. Go ahead.
Yeah.
Well, I was just wondering, in all these because we're talking about one protein that used to be viewed as performing one function, but now we know that they can perform different functions even though they're the same protein. And a lot of that has to do with the folding. Have they come up with like equations and formulas to indicate how many possible combinations or foldings there are for a particular protein, or is that something you just have to experiment on and discover?
That is the biggest golden calf in the industry right now, coming up with those. And they recently had an advance that put us like 50 years ahead, I think they said, in protein folding. I think Google came up with it. They used artificial intelligence, but figuring out how proteins fold is something that we didn't think we were going to be able to do for like 50 years. It takes immense computer power to do that. There are no formulas. We have some ideas about what happens. There's two different ways the bond can rotate. One is called phi and one is called psi, hence sci-fi.
Wi-Fi.
Yeah, but figuring that out for a protein is just incredibly complex. But Google,
would you say it's deterministic but complex, or that it's not deterministic at all?
There's a set of physical kind of parameters that you can conceive of that kind of give you an idea of what a properly folded protein, or what a stable result for a folded protein should be. And I forget, I have a textbook on this type of thing, but I think the textbook I have, it lists like 12 or so parameters that are kind of relevant. Either way, the exact number isn't necessarily important, but the thing is, right, when you consider something like the number of bond rotations that can occur, between amino acids. So, like Speed was mentioning, you have phi and psi. One of them pertains to the carbon bond of the carbon bonding to the amine, and the other one pertains, if I'm mistaken, to the carbon bonding to the carboxyl part of the amino acid.
Yeah.
And the thing is, right? Okay, so one amino acid corresponds to two angles you need to consider, which are psi and phi, and how these angles behave relative to one another, right? And remember, each one of these can range from 0 to 360, but they won't necessarily do that because of certain chemical features that amino acids have, and that it limits how much rotation you can have. But okay, fine. The more important aspect is just for that one parameter, right? Most proteins, and Speed, correct me if my number is wrong on this, the average length of some proteins is like usually hundreds, sometimes thousands of amino acids.
Yeah.
So if you take even the low end of that number, right? If you were to say, okay, let's consider a protein with 700 with 700 amino acids, right? Then, ignoring every other parameter that you could contrive of, and you just consider, okay, let's consider the bonds of each amino acid, each psi and phi bond of each of those, of each of these amino acids relative to how they're connected. You have, 2 times 700, you have 1,400 different angles now that you have to compute, and you have to consider how the contribution of those angles, how the proportionality of these values influences the stability of your protein, because it could be the case, right, that you have um, cyan 5 values for amino acid 23 that are really nice. Okay, good. That's really nice to see. It's not nothing too crazy is happening, but crap, where it is, how this molecule is rotating in space, which is kind of what those things describe,
Mm-hmm.
relative to amino acid 500. Oh well, that's not very good at all, because now there seems to be kind of a clash, if you will. There's electronic repulsion between this side chain of amino acid 23 and this other side chain of amino acid 500, and in the protein chain there appears to be this kind of the angles, the way they're relative to each other, seem to say that, okay, there might be this might cause a very high energy situation in this part of the protein, which is very unfavorable. I mean, the point of this all is to kind of illustrate, even with one parameter, right? Even with one parameter, the size of the protein itself, the fact that there's so many amino acids that make a protein, and the fact that amino acids are not something which are just like point particles. They're not something which you can just draw, which you can just put a dot and just say, oh well, that's what the structure is like. It's just a point like structure. No, these molecules, they're three-dimensional objects, right? They exist in the three-dimensional world. You can define them with a proper set of coordinates, and you also have to consider the rotations of those molecules, and also the fact that obviously these molecules react, right? They have electric fields. There's parts of that molecule where where electron density is high, and the molecule feels very satisfied, very negatively charged, and parts of it where electron density is not so strong, and the molecule feels very strained and very electropositive. And it's just the fact that proteins or amino acids are three-dimensional makes things enormously complicated.
They have something called the Ramachandran plot.
Yeah.
That, let's say I've got a picture of here.
Yeah.
And it's
cyan 5 bonds relative to each other.
And that's also extremely variable. And let's see, put that up. And that is the, let's see here, that is the 5 psi angles, basically. Here's psi between the two carbons, and then phi between the carbon and the nitrogen. So that's how they do that. And then a Ramachandran plot is, where's my Ramachandran plot, Loki? Loki doesn't know where it is. Here it is. Okay. So then they plot those angles, and you can see that it's not deterministic. Okay, it's somewhere in these, these groups, depending on how the hell is this? I'm not sure exactly how the plot's done anymore. But anyway,
you're plotting essentially. So essentially, what you're doing with a Ramachandran plot, and there's a better, I'm sure there's a better picture you can find with it. But essentially, what you're doing is you're saying, I think that plot is actually considering, what is the scale, minus 180 to 180. So what it's describing, right, is it's describing the orientation of a bond relative to the orientation of some other of the orientation. Okay, let me re-say that. God, Jesus.
It's psi plotted against phi,
right?
Depending on which angle, and they show what the dense areas are what the preferred folding is for the protein, right. and so it'll kind of bounce around between these areas, you know. And I'm not sure if you can see my cursor or not. You can't.
Yeah. What you have to remember too, right, is that when you obtain a Ramachandran plot for some kind of peptide or protein or whatever it is you want, right? What you're doing, right? What you're capturing, whatever angles you happen to have obtained, you've obtained those values for that protein at one moment in time, right? That doesn't necessarily reflect the nature of the bonds and how they're positioned relative to each other of the entire protein.
Yep.
That, as you said, there can be variation within that plot, and within how close or far apart some of these bonds get to each other.
And he points out how it's even worse a little later on, but I'm not sure how far we're going to get tonight with this. Somebody want to read the introduction of part 2?
Why has it taken us?
What? No, much of.
Much of what we know about the cell's organization derives from snapshots of fixed, stained, or desiccated biological samples obtained by conventional microscopy techniques. A representative example is shown in Figure 1. Historically, the interpretation of images of this kind naturally led to an understanding of the internal architecture of the cell in terms of clearly delineated, neatly compartmentalized structures that closely resemble machineries. These permanent structures were essentially assigned functions to make sense of their role in the overall economy of the cell, which, in accordance with the MCC, was viewed as a factory with highly specialized compartments.
Yep.
But how are these structures that constitute the cellular architecture formed and maintained? What is it that determines their different shapes and sizes, as well as their respective locations and functions in the cell? For decades, the basis of our understanding of macromolecular order was the principle of self-assembly. Self-assembly involves the physical association of molecules into a static equilibrium structure in the absence of an external energy source. It is driven by local stereospecific interactions between the aggregating building blocks, which remain unchanged throughout the process. Essentially, what that's saying is how these building blocks are being packaged relative to each other. That's really complicated phrasing. As the properties of the resulting structure are determined by the property of its parts, resulting structures are determined. Self-assembly can be regarded as an extension of the central dogma of molecular biology, bringing us from the realm of linear information to the realm of protein assemblies. Classical, well-studied examples of self-assembly include viral capsid formation and ribosome biogenesis. However, self-assembly is not the only theoretical principle that can be linked to explain the spontaneous generation of macromolecular order. There is also the principle of self-organization. Self-organization refers to the collective behavior of molecules when these interact non-linearly to generate a dynamic, far from equilibrium structure, sometimes called a dissipative structure, which maintains itself in a low entropic, steady state by constantly expending energy and exchanging matter with its surroundings. So, while self-assembled systems are closed, as the material constitution is conserved, self-organizing ones are open, as they rely for their preservation on the continuous replenishment of the material that composes them.
And that's also a good model for the autopoietic nature of the cell, that it self-assembles and self-organizes, and it is constantly in flux and needing energy from the outside, and that's why we die. When it quits doing that, everything kind of falls apart, and it just, it melts. So it's holding itself in this form, this self-organization, and as soon as the energy stops coming in and the system breaks down in some critical way, everything just kind of melts, and then we have mushy. Yeah.
Well, what begins happening, right? I mean,
Well, you shouldn't eat wieners that have been in the fridge too long.
speak for yourself.
Okay. Let's see. Oh, you were going to say something?
Yeah, I mean, part of it is the fact that, you know, as organisms grow over time, right? The process of mitosis is not perfect. It tries to be. It genuinely does try to be as efficient as it can. But the unfortunate truth of, you know, genetics is that mutations happen, and over time, over the course of an organism's lifetime, proteins do accumulate damage. Now, sometimes that is metabolic damage. That's the, you know, it's the production of, you know, what are called ROS species, so radical oxygen species that wreak havoc on the cell and cause damage to DNA. But sometimes it's not the result of chemical damage necessarily. Sometimes it's the result of mutations occurring in the genome, and those mutations over time they build up, and after a while, right, the protein that was, you know, really good when you were an infant has, you know, has kind of aged over the years, and it doesn't necessarily, you know, do as well. It doesn't do as well as it used to, and that might be because certain domains of that protein were, you know, far from optimally mutated, and that causes changes in the structure and what have you. And so it's it's not necessarily the fact that, you know, that the energy just stops existing, right? It's not like, you know, we're eating so much or we're consuming so much of some energy source that that it's going extinct, right? Sometimes it's the fact that how we process that energy, the ability of our body, the ability of an organism to extract the energy as efficiently as it was, you know, as it was able to, you know, when it was, you know, an infant or a baby, right, has decreased. And as a result, you know, that this process of degradation, or this process of, you know, this slow and gradual decline, you know, eventually will lead to a point where the organism can no longer support itself because the damage has, you know, gone too far.
Dissipative structure is a really good word. Maintains itself in a low entropic, steady state by constantly energy and exchanging matter with its surroundings. And then a far from equilibrium dissipative structure, in the absence of a steady supply of energy, reaches equilibrium and dies. And then self-assembly and self-organization tend to lead to similar observable patterns, albeit through totally different means. The difference being that those generated by the former exhibit static stability, whereas those generated by the latter exhibit dynamic stability, which they refer to as metastability, which is also a good keyword from all of this. So what else we got here on this page? Let's see. Recent technological innovations have changed all of this. The development of in vivo microscopy techniques using genetically encoded fluorescent tags of individual molecules has provided new insights into the spatio-temporal configuration of the cell. Perhaps the most surprising discovery that has emerged from these studies is the unexpectedly high degree of dynamism observed for a wide range of macromolecular structures. That is a fucking mouthful of marbles. It appears that many, perhaps most subcellular compartments are more appropriately described as dynamic, self-organizing, steady states than, and here's one, as static self-assembling machineries. So that's kind of an interesting key concept. And we have a picture of a green algae.
Yep. Essentially what they're saying, and I'll use a mechanical example, okay, right, is that it's kind of like a ball that's sitting on a hill, that ball is perfectly happy sitting where it is and not moving worth a damn. But if you give that ball a little push, right, especially when it's on the top of the hill, yeah, that sucker is going to fly. That sucker is going to move, really, really quickly and gain energy going down that hill, and it'll do its own thing. In the same way, right? Like proteins, you run into this kind of interesting little problem called what's called the Leventhal paradox, where um, where the free energy of an environment. So that means the amount of usable energy in a system that is that a the amount of energy in a system that's usable by a molecule or whatever sometimes is not sufficient to tell you about how that structure will fold or how that structure will finalize. Because, kind of like we were saying and talking about earlier with the Ramachandran plots, um, you can have a lot of very, very similar structures or very, very similar chemical interactions that are close by and that are still relatively stable, right? That are what this is calling semi-stable. So it might not be the most stable the molecule could be in, but it's stable enough that once it reaches that conformation, uh, the free energy of the environment is not sufficient to make it fold anymore, or to make it so that it would rather adopt an energy that is in fact more energetically favorable for that compound to be in.
Any comments, Scott?
No, I'm just soaking it in.
Okay.
I feel like I might be going too hard into some of this, so just tell me to shut up if I'm making things a little too complicated.
Yeah, we can kind of follow the outline of the paper. That'll help.
Well, I'm just raising examples that are relevant.
Yep, you are. Particularly Leventhal paradox. I might put some words over this later. I don't know. These transient metastable systems are sustained by the incessant flow of energy and matter passing through them. So that is a theme that keeps coming up. That there's this constant influx of energy and matter passing through, in and out, and displaying different recruitment probabilities. So he uses an example. The mitotic spindle of eukaryotic cells is one such example. The spindle is an ordered array of microtubules, associated proteins, and chromosomes that forms during cell division and which distributes the duplicated genetic material to the daughter cells, how cells divide, basically. Owing to its remarkably stable, often crystalline appearance in cross-section of cells undergoing mitosis, the mitotic spindle is often characterized as a fascinating protein machine. Let's see here. Maybe we can skip ahead. Research has shown that the spindle is actually a self-organizing system displaying high degrees of flexibility and robustness. So it's not so much a crystal as it is a self-organizing system. Architecturally speaking, the microtubules that compose the spindle are constantly polymerizing and depolymerizing, which is an interesting thing about microtubules. They're on one end, they're growing; on the other end, they're destructing themselves. So they're very, very dynamic, and that's what bothers me about the little man walking on the microtubule. Microtubules are like creating themselves on one end and disappearing on the other end. It's kind of a funny looking highway. If you could imagine if your freeway did that, it would really piss you off. Let's see.
Well, not if it lets you get if it lets you get to work a little bit earlier.
Yeah. Let's see. I
had early. Sorry.
Yeah.
Or later.
Yeah. Or later. Yep. Okay, let's see. The traditional view, as a consequence of these findings, the traditional view of the mitotic spindle apparatus as a molecular machine which is built through a defined, irreversible set of instructions is gradually being replaced. So, hang on a second.
And that's not just Nicholson speaking, unless he's not quoting his own paper, is he?
That is another paper that he's referencing from Duncan and Wakefield,
Okay.
2011.
As long as it's not Andrew Wakefield.
Who's that?
Oh, he was the doctor that lost his license or his whatever for falsifying information about autism and vaccines.
Oh,
Yeah, he's one of the fuckers that pushed this whole, vaccines cause autism bullshit.
well, he could have done some good work. I mean, even John Sanford did some good work in the past.
Wow. Yeah, it's hard. It's really hard to
Zing.
envision that.
Let's see. Self-regulating dynamic structure where multiple pathways of microtubule generation are spatially and temporally controlled and integrated, constantly talking to one another and modifying the behavior of their MTs microtubules in order to maintain a flexible yet robust steady state spindle. So there's some new research on that indicates not so machine-like. Okay. Somebody want to read?
Sure, where are we?
We're on It Has Further Been Suggested.
Okay, yeah. It has further been suggested that that not only the mitotic spindle, but the entire cytoskeleton is better characterized as a metastable flux dynamically responding to changes in its environment than as a static macromolecular construction. Despite the connotations of the word skeleton, the skeleton is not a fixed structure whose function can be understood in isolation. Rather, it is a dynamic and adaptive structure whose component polymers and regulatory proteins are in constant flux.
Yep.
Self-organization appears to be similarly crucial for intercellular membrane compartments, such as those involved in the secretory transport pathway, in which proteins targeted to the cell's exterior are transported from the endoplasmic reticulum through the Golgi complex to the plasma membrane. Although the compartments of this pathway have traditionally been regarded as static structures, the recent tracking of resident and cargo molecules through the pathway using in vivo microscopy has revealed that they are in fact constantly exchanging material. The Golgi complex, for instance, resembles the mitotic spindle in that its stability is a consequence of the balanced turnover of the molecules that flow through it. So that's kind of interesting. Given its fluid nature, its architecture can be modified by manipulating the influx and efflux of the material passing through its compartment cristae. We now know that the inhibition of traffic from the endoplasmic reticulum leads to the dispersion of the Golgi complex into small vesicles.
That is interesting.
Yeah, that is very interesting. Whereas blocking the transport of vesicles that bud from it results in its enlargement. Although there is still considerable disagreement over how the actual traffic occurs, specifically over whether the Golgi cristae themselves progress or mature along the pathway, or if it is only their cargo that gets transported. What seems clear is that the Golgi complex is a self-organizing, steady-state organelle. More broadly, live imaging techniques are unveiling the striking dynamicity that underlies the stability of intracellular membrane compartments of both eukaryotic and endocytotic pathways.
That was endocytotic.
Yeah, mitotic.
Psychotic.
I'm getting psychotic right now.
Endocytotic.
I can take over reading if you're getting.
No, I got it. I got at least one or two more paragraphs.
We got to share with Scott.
What do you like to read, Scott?
I'll start with our understanding.
Yeah.
Our understanding of the eukaryotic cell nucleus is also becoming radically transformed. Far from being the static, crowded, gel-like structure described in textbooks, the nucleus is extremely dynamic and surprisingly fluid. Most of its proteins are highly mobile, stochastically moving about the nucleoplasmic space, contingently interacting with one another and participating in different nuclear functions, such as chromatin remodeling, transcriptional activation, ribosomal RNA processing, and DNA repair. The dynamic interplay between nuclear proteins results in an ever-changing yet globally stable architecture within which nuclear processes take place. The nuclear architecture includes a number of morphologically and functionally distinct compartments, such as nucleoli, I'm going to try this one, Kagile bodies, and paranuclear specks that are maintained in a state of perpetual flux. Did I pronounce that right? Kagile or Kagile?
I think it's either casual or casual. Casual.
We could go to the pronunciation Google-y thing, but we won't.
We won't, okay.
Okay.
In a state of flux, perpetual flux by the constant exchange of their resident proteins, which also transiently associate with the chromatin. The latest research on these subnuclear membraneless organelles strongly suggests that they are better conceived as liquid-like droplets than as a solid core shell structure. They have a spherical shape, they fuse together, and their molecular constituents are constantly undergoing fluid internal rearrangements.
Yep. The interesting thing here is that subnuclear structures they don't have membranes, and they self-organize and they're very liquid. And one of them is the nucleolus, did I say that right? Where RNA is basically the RNA is for the ribosome, and a number of other things are created. And what else is there? There are a few other really important proteins that are created there, but the ends of chromosomes tend to cluster around this little group called the nucleolus, and it's kind of like a little machine. Did I say machine? Fuck.
Damn it, Pete. Son of a bitch.
Fuck, fire me. Anyway, it's kind of like a little, I was going to say factory. It's a little, it's just a little cluster of constant transcription going on right there. Anyway, God, I'm fired.
If ever this sees the light of day and Otangelo watches it, he's gonna
jump on that
comment randomly on the video. It's like, oh, Pete believes the cell is a machine.
Said.
Well, remember the idea of the cell or the parts of a cell as machines is fine as far as it goes, and it's true in a sense, but you lose information if that's all you do is look at it mechanistically. There we go. So saying it's like a factory here or a machine there is fine. It's just that it's more than a machine. And sometimes referring to it as a machine works, sometimes not.
It's like a hurricane. It's like a little cluster. Like you've seen hurricanes move across the Atlantic. Mm-hmm. and hit you down there, Immutable. Hit you right in the ass. And these little things appear to be structured if you look at them from space, from the satellite, and they whirl around and they dissipate. Anyway, the nucleolus is like that, except it sticks around for quite a while in the cell.
And nucleolus is like I swear to God,
like a hurricane. And if we had an atom bomb, we could probably move it, right?
Well, if it takes that much energy, the cell isn't going to be fucking alive, but okay.
Somebody came up with that idea.
Yeah, I know who you're referring to.
Okay. So the self-organizing.
Okay. Let's see. The self-organizing nature of the cellular architecture has far-reaching theoretical consequences. Most fundamentally, it leads to a view of the cell that is completely at odds with the MCC. For one thing, it dispels the notion that the information that specifies the spatial organization of the cell is somehow encoded in the genome.
There we go.
Huh?
There we go. This is important.
Is this part of that non-genetic heterogeneity? Is it the concept?
Sort of, yeah.
Okay.
Yeah, this actually kind of relates to what's called the I swear to God, I have to say this. What's called the signal hypothesis, if I'm not mistaken.
You're fired.
You're fired. and what it says essentially is that there's certain signals, if you will, within the structure of proteins that specifies their location within a cell. And one of the ways that it accounts for that essentially is that essentially it has to do with the fact that the ribosome uh, translates this protein, near the area of interest. That's a very kind of brief explanation of what it is, but if I manage to think of the video, I will find the series on it and link it.
Okay.
But now I lost my train of thought. Let's see.
You were reading some paragraph.
Yeah, there we go. Self-organization generates order in the absence of an external template or global plan. Genes specify only the primary sequence of macromolecules. The architecture of the cell, for the most part, arises from the interactions of numerous gene products with other cellular components. Genes are important, to be sure, but they do not set in motion a unique chain of events that produces the organization of the cell. As the use of the term information sometimes misleadingly suggests, rather gene products are released into a cellular milieu that already possesses spatial structure.
Yep.
And they exert their influence under the physiological constraints of the existing order, much of which is shaped. Under the influence of the physical constraints of the existing order, much of which is shaped by pre-existing self-organizing processes.
Yep, and that's the mind-blowing thing about life, is that the zygote that comes from your mama has the structure of that is what allows the organization to the organism to start to organize itself. With just the genome, nothing would happen. So it's more than a blueprint. There's an existing context and structure that's passed from one generation to another generation that allows life to keep going. And that has been going for a long time. And that is very interesting when you understand that it is not a damn blueprint. It is a cluster of information that, when in the context of an already existing cell, continues on into the next generation. And it continues on in a very, very slightly modified form every time. So life is constantly unfolding, modifying itself, and unfolding again and again and again and again. And it's remarkably stable from, like your kids are still human.
Well, are they going to become like werewolves or what the fuck?
I mean, yeah, they don't turn into a banana or turn in, what does Kent Hovind say? You don't want a banana doesn't turn into a pine tree, something like that. But anyway, but it does change in every generation. There's always that variation. So I mean, when you think about it in those terms, you see life, it's completely all connected in this vast network. It's absolutely amazing. And we haven't even
guys.
What's that?
I'll be back in just a second.
Okay. So
I'll bring this up while he's gone, but it does kind of make you think about it, right? In the sense that if the structure and function of some of these
what did I do?
What the hell did you do?
I don't know what I did.
If the can you do like Control Z or something, or what the fuck?
No.
Oh, there we are. You're back. No, we're up. We're up from that. Yeah, down a little.
There we go.
Yeah, we're at the unlike the engineered macroscopic. We'll wait till he gets back to do that.
Okay.
What I was asking though is if the structure and function of the nucleolus, right, is constrained and defined by, a set of existing physical parameters, right? Then for the origin of life, right, the only way life could have completely, could have finalized in its development is if there was some way or there was some process in which, these kind of physical constraints were being consistent as as protocells divided and mutated and so on and so forth, right?
Yeah, it's
Right.
like you can project the movie forward and look at, this cell is going to become slightly different and it's going to turn, and follow that line into the future. But you can also project backwards and look at how it arose from very, very simple systems, far simpler. And that is in fact what we see when we look at prokaryotes. Or ribosomes are less complex than eukaryotes, and even though they have granted been evolving just as long, so they're not what they used to be. But you have to look at that development. When you look at abiogenesis, you're projecting all the way back to rocks.
Yeah, you're projecting to something very, very primitive and very primordial.
Yeah. And somehow this self-creating, self-perpetuating, self-organizing system got started. The match was lit. And once it started, it took over the earth like pond scum.
All right. Let's see.
Going on.
Engineering structure. Let's see. Do we want to stop once we've finished table 1, or what do we want to?
I think we want to. Should we run 2 hours?
I think what we'll do. So you've read this paper, right?
Yep.
Speed. Where do you think is a good place to stop for this particular session?
Number 3 is protein complexes, molecular machines, or pleomorphic ensembles, and I am very excited about that. And we could probably, so if we go through table 1, we could stop there, and we could hit that at another installment.
All right, so I'll read until we hit the table 1 part.
Okay.
If you want to stop after a paragraph or so, then we can and discuss that.
Okay.
Unlike the engineered macroscopic structures that we commonly build, biological structures are dynamic and self-organized. They sculpt themselves and change their own architecture, and they have structural building blocks that constantly come on and off. A description of such structures defies current construction mechanical frameworks.
Okay. Traditional mechanical frame. Okay, we're up there.
Indeed, no machine self-organizes by autonomously exchanging its material constitution in order to maintain its architecture in a dynamically steady state.
Take that off.
Yeah, fucking shit. Yet this is precisely what happens in every cell. But why do cells favor self-organization over self-assembly as the main mechanism for creating their architecture? Would it not make more sense for a cell to build static equilibrium structures that do not require a constant expenditure of energy to maintain them? Although self-assembly is a more economical and efficient means of producing durable macromolecular structures of great complexity, the viral capsid is a conspicuous example. The resulting structures lack morphological flexibility and do not lend themselves easily to modifications. The advantage of a self-organizing architecture, despite its huge energetic cost, is that it confers a great deal of plasticity without compromising on stability. It allows cells to respond rapidly and adaptively to external perturbations and other critical events that would otherwise jeopardize their systemic integrity. Overall, recent research on the cellular architecture demands that we look more carefully at what we have previously assumed were well-defined structures and reconsider them as stabilized processes. Because processes are temporally extended, it follows that they can only be understood by giving time due consideration. And herein lies the problem. The methods traditionally used to probe the interior of the cell conceal the dynamic nature of its architecture, because they have to incapacitate it in order to render it visible. Yet to study a cell frozen in time is already to approach it artificially as a static machine-like object, rather than as a fluid system that it is in reality. The structure of a machine, after all, can be grasped in abstraction from time. as it is not constantly changing, whereas the structure of, say, a whirlpool or a stream cannot. This explains why, when we have started using techniques that allow us to examine the cellular architecture in real time, we have found that many of the cell's compartments and organelles are not fixed machineries at all, but stable macromolecular fluxes.
That's an interesting term, stable macromolecular fluxes. I have trouble with that.
He pretty much almost used your hurricane analogy.
Yeah, he did. Yeah,
He did.
he did. But I have read this paper, so I cannot complain.
Was that an intelligently designed alliteration?
Yeah. Which one?
Was that an intelligently designed analogy?
Oh,
you gave earlier?
No.
More broadly, the transition from a structural to processual conception of the more broadly, the transition from a structural to a processual conception of the cellular architecture implies shifting our attention from matter to form. Due to its dynamic nature, what persists in a cell over time is its form, not its matter. The individual molecules that make up a cell come and go, but its overarching organization remains. Accordingly, if we are to grasp how a cell operates, mapping out the network of spatial and temporal relations that exist between parts as is, if not more important than characterizing the parts themselves. Actually, wait, I misread that. Let me read that again. Accordingly, if we are to grasp how a cell operates, mapping out the network of spatial and temporal relations, rather than exist between its parts as it is, if not more important, relations that can exist. Yeah. If not more
important than characterizing.
Yeah, okay. I misread. If not more, if not more important than characterizing the parts themselves, the need to adopt a non-reductionistic stance is further intensified when we bear in mind that the self-organizing processes, which, as I have shown, underlie much of the cellular architecture, force us to focus on the systemic patterns in collective behaviors, rather than on the properties and structures of the single molecules, which would suffice as an approach if the cell were primarily self-assembling and its order was ultimately encoded in DNA. The main differences between the two conceptions of the cellular architecture I have discussed in this section are summarized in Table 1.
Okay, so Table 1. So we have static structure versus stabilized process, which is what he's calling it. Self-assembling versus self-organizing, closed system, open system, at equilibrium, far from equilibrium, genetically encoded, emergently generated, economical but inflexible, and this is costly but flexible, and temporally abstractable, temporally non-abstractable, what the hell? Amenable to reductionism, not amenable to reductionism. So very difficult to come up with a reductive diagram of, the cell. So anyway, I think we could stop there for, but there is this picture, which is, I think, what is this on? Wiring diagram.
Metabolic network.
Yeah, the metabolic pathways. And I mean that if you were to actually detail what's going on, this would be one solid pile of
mess.
It looked like snow on your TV screen, if you remember that. Back when we had snow on TV screens. Do you remember that immutable?
Sort of.
Huh? Yeah. Yeah, that's what used to happen at 12 o'clock every night. There'd be like this little pattern and then snow. And you'd wake up and there'd be fucking snow on your TV screen because they didn't broadcast at night.
And it was kind of featured heavily in the movie Poltergeist.
Yes.
J Nicholson and them, because the daughter was sucked into the TV screen or something.
Right.
Yeah. During all that static. But anyway, I remember that snow on TV.
Yeah, these diagrams are alluring until you realize how fragile they are and how they change. And the next section that we will get into is the pleomorphic protein complexes, molecular machines, or pleomorphic ensembles, which is really a good one, and I really like it. And I think we'll stop right now.
Vocal cords fucking shit.
We could broadcast this part of it, but